All right, thank you, Caleb. Looks like the scaffold quant is coming along nicely. All right, let's move to a presentation from one of our guest speakers. Next, we're going to hear from Mak Saito. Mak is a senior scientist in the Marine Chemistry and Geochemistry Department at the Woods Hole Oceanographic Institute. And Mak is presenting a talk entitled Complexity in Ocean MetaProteomics, 20,000 Spectral Counts Under the Sea. So with that, take it over, Mak.
Great. So yeah, thanks for the opportunity to speak today. As mentioned, I'm a scientist studying the oceans, and I went back and looked to see when I first started using Proteome software. It looks like about 12 years ago. This is a picture of one of the field environments we've been working on, McMurdo Sound of Antarctica. It's actually a really beautiful place with some whales here, and this is sea ice and Erebus volcano in the background. Let's see if I can change my slide here. So yeah, today I'm going to be talking about how we're trying to apply proteomic technology to the marine environment. And apologies to Jules Verne for this changing his title. But our emphasis has been primarily on wet lab method development and field sample collection to try and bring proteomics as a tool to understand how the oceans are affecting global carbon cycling and how they work both biologically and chemically. And there's a number of major facets of our research. Sort of our bread and butter is microbial proteomics, probably very similar to a lot of the experiments that other people do for model organisms, except we work with a lot of non-model marine organisms, bacteria, archaea, eukaryotic microbes. Because a lot of these microbes are recently in culture or difficult to culture, we're often using custom genomic and transcriptomic data sets for our peptide spectrum mapping. Based on these data sets, then we learn things that we can go out into the oceans and try and understand how natural samples in the oceans work for their proteins. In other words, the metaproteomics of the natural environment with the entire community mixed together in our samples. And a lot of this is global metaproteomic research. That involves custom metagenomic and metatranscriptome data sets for our PSMs. And then we've focused pretty heavily on doing targeted metaproteomics using stable isotope labeled peptides, using multiple reaction monitoring and parallel reaction monitoring. We also do a lot of metalloproteomics. I won't talk about that today, but basically it's a tool that does native 2D separation and then does metal and protein dual mass spectrometry characterization. And there's a recent study here if you're interested in learning more about that. And then this generates a lot of data, far more data than we can use. So we've been building data sharing capabilities, in particular the Ocean Protein Portal, which I'll talk about. And a lot of these pipelines, almost all of them have use scaffold products, proteome software products, and we really appreciate that product line because we're so focused on doing the mass spectrometry and the extraction development and the field logistics. Having sort of a really high quality informatics capability that's adapting and growing really has been very useful for us, so we can focus on all the other complicated things we're trying to do as well. And here's just a picture of Woods Hole. If you haven't been here before, it's on Cape Cod, Massachusetts, US. So I often get asked in these ASMS type talks if we go out on the oceans, and so yes, we do go out. Here's a picture of two of the ships that Woods Hole has run over the years. This is the Neil Armstrong and the Nor, which has now been replaced. Yes, we go to sea often from 2 weeks to 70 days all over the world. I've been to every ocean except the Indian Ocean at this point, and we've even managed to bring Brian Searle, the co-founder of Proteome Software, out to sea with us. I would talk to him at meetings, and he would often say how complicated our science was, and after I think many years of badgering him, he finally agreed to come out and take a look and see what we're all about. And so it's been an exciting new collaboration for us. We've just a bit of an aside, we're also developing robots to try and help increase our ability to get samples. The oceans are huge, 70% of the Earth's surface. We're trying to understand how they work and how they're changing. And so collaborating with engineers to go out and get high quality omics samples. This is several hundred liters filtered through a filter here. Using these robots is a big improvement in getting samples back for us. And this is actually one of the cruises that Brian came on with us off of Bermuda. Oops. So yeah, here's just an image of breaking through the ice in Antarctica as well. A lot of these microbes are not very well characterized. This is an example of this, the Southern Ocean. It's very hard to get to. These cruises tend to be 2.5 months long. We're studying a lot of if you see some of the brown down below here, that's sea ice algae that are living on the bottom of the ice. So it's an important ecosystem that is changing rapidly as well. Okay, so I'll talk a little bit about our laboratory experiments just to set the context for our field work as well. We often do lab experiments on microbes to try and find new biomarkers for environmental stressors. And this is an example of a study we did a while ago where we took some marine algae, diatoms, shown here, and we starved them for vitamin B12. And these are simply replete on the bottom axis and B12 limiting on the vertical axis, and proteins shown here. And you can see that one protein, this one in purple here, was far more abundant under B12 limiting conditions than without. And this is what actually happens in a lot of our experiments. If you take a key nutrient away from a microbe, it'll have a very strong adaptive response where it, in this case, produce a transporter, produce a protein of unknown function that we then characterize through overexpression to show that it was a transporter. And now that allowed us to have a new biomarker for B12 limitation or B12 stress in an important marine alga in the oceans. One of the things I want to point out is that we're often looking for very big signals, because when you have a unicellular organism and you starve it for something, it will make a ton of a transporter or a chaperone or an enzyme to respond to that deprivation. And so that gives us really nice big proteome targets to go after. And so that's one reason I think this method seems to be working really well in environmental settings. We've also done RNA to protein comparisons. We often hear how RNA and protein don't correlate, but we actually find that they do correlate for some of these environmental biomarkers or stressor proteins because the expression level is so high. So the RNA and the protein kicks up really high, and so you get a very large signal for both if you sample in the right time window. And so with this knowledge of biomarkers that we're gaining from our lab studies, we can go out into the environment and try and see what's happening in the real world. And there's a lot of challenges, though, in trying to do this. One of them is sample collection, getting out there. You need ships. You're trying to get samples from a dilute environment. The analysis is very complicated. The samples are complex with many organisms mixed together. The data analysis is complicated, and of course, then sharing the data is really important because we need to get this out to the community. One of the techniques we've been working with in recent years is 2D active modulation to get really deep into our proteomes. And this is an 8-hour nonlinear 2D gradient that has dual traps and a high pH, low pH set of columns. And it's effectively like 10 or 15 HPLC runs happening from a single injection, but that allow actually very narrow peak widths, so we can get pretty deep into the proteome that way. And that's been working really well for us to get into our samples. You can see this is a 1D versus 2D comparison, and we get roughly 4 or 5-fold more peptide IDs. And here's an example of what some of these biomarkers look like in the ocean. This is actually MRM data using a triple quad, but the targets were built off of discovery using spectral counting. And in this case, we're going from Hawaii to Samoa on this map here, and Hawaii is on the northern end and Samoa is on the southern end, crossing the equator. And you can see nitrogen stress proteins are more abundant in the north and disappear in the south, and the opposite happens for iron. And then we can take these data sets and then interpolate them to make what we call ocean sections. So this is now the north and this is the south. And as we go from north to south, we go from a region of nitrogen stress to a region of iron stress that corresponds to where those nutrients are in the oceans. And so this was really, really exciting to a lot of us because it showed us that these biomarkers really can see changes in how the ecosystems are functioning and how they're responding to their chemical environment. I should point out that there's nothing down deep here because this is hundreds of meters and there's no light down here. So these photosynthetic organisms don't exist down deep. And one of our more recent observations is that as we went down into that dark zone in the ocean, the most abundant protein turned out to be an enzyme that had 23 atoms of iron per complex. So this was a really big shock to us because, as I mentioned before, the upper ocean is iron limited, but then you go down deep and literally the number 1 and number 2 most abundant proteins appear to be iron proteins. And this is consistent with when you take the organism that this comes from and you grow it in the laboratory. Indeed, inside the proteomes of those laboratory organisms, those proteins are most abundant as well. And what we think is happening is that this organism is living off of oxidizing nitrate with oxygen, and so it covers its membrane with this enzyme, just waiting for nitrite to come down, and its organic matter degrades from sinking plant growth in the upper water column or algal growth. It's ready to pounce on those chemicals that are coming out of it and create energy, and that's why it's basically covering itself in this. But this is an interesting example. Basically, you know, we're looking at the top 2 most abundant proteins in the proteome and able to write a story about it, and we have 70,000 more to go in terms of describing that story. And one of the things that we've realized is that normalization in metaproteomics is actually fraught with challenges. And it's something that metaproteomics people can debate at no end. But you know, the reason for this is that in the laboratory experiments, we often use normalized spectral counts or normalized spectral abundance factors, or as Gail just talked about, you could normalize the proteins if you're targeting. But in our system, the biological matrix is actually changing with depth, and also there's also the potential for organic matter in the form of non-tryptic peptides from degrading organic matter that's sinking and other organic molecules. And so here's actually the sum of spectral counts as you go down into the water column. And you can see it's really changing quite dynamically. And if we normalized here in blue to roughly 5,000 spectral counts, and then you can see what the profiles of 4 different proteins look like, you can see that the difference between the normalized and the total spectral counts really changes quite a bit depending on where you are in that vertical profile. So one has to be careful about normalizing and question whether or not you want to normalize at all in this context. But the end result is that, you know, for some of these data sets that we're now looking at have roughly 80,000 protein IDs in them, and amazingly, Scaffold's able to handle that as we map to our metatranscriptomic and metagenomic databases. And that basically means that if we have that many IDs, we're getting that many ocean sections, like I talked about before. So here's one section of this more recent expedition we did called Proteomes, and it's a TON-B iron transporter, and it has this really neat distribution pattern right below the sunlit layers of the ocean, and changing as you go farther south. And so we think this is a really valuable record of the ocean's metabolism in space and time. And it could be useful for both basic research and detection of environmental changes, and also for education, for teaching about the ocean or biochemistry or microbiology. And so to share this data, we've been developing a web portal called the Ocean Protein Portal, where you can go in and type your protein of interest. Here's an example of looking for a phosphate transporter, and you can create an ocean section in real time. And this is all currently spectral count data, but it really could be any units that people want to deposit, and it's meant as a resource for the oceans community, for people who are generating data, to share it with people who are interested in enzymes or transporters or microbes, to be able to quickly access more information about a protein of interest. Let's see if I can go to the next slide. There you go. And one of the key questions when you start working in metaproteomic samples is who is making the protein. And so one of the key ways of thinking about this is to figure out if the tryptic peptide you're studying is unique to a particular taxa or not. And so we have a tool called Metatrip that we just released a version 2 of, that lets you dig down to the taxonomic level of that tryptic peptide. There's a parallel product called Unipept, and the difference between the two is that Metatrip really allows you to have specialized sequencing assemblies, so MAGs and single amplified genomes and metagenome amplified regions. So whereas UniPept is primarily curated model organisms in it, and so that's why we had to create this product. But it allows us to dig down and figure out whose peptide is whose. Let's see. And so more recently, we've been exploring using DIA as an alternate means of quantitating this data. And this is in collaboration with Brian Cyril here at Proteome Software and Institute for Systems Biology. And it's been pretty exciting so far, where we're taking our 2D outputs, mapping to get our PSMs, using our metagenomes, and then creating a list of discovered peptides, and then using PROSIT to generate our spectrum libraries, and there'll be more talks about that in this session later. And then using scaffold DIA to take our injection data, be it one injection or gas phase fractionation, 3 and 6 phase gas phase fractionation, to generate our peptide and protein reports. In this instance, we've done a 500 to 1100 m/z precursor range with 24 m/z staggered windows for one injection, or 8 and 4 m/z staggered windows for the multiple injections. And here's the performance where we have the single injection actually does pretty well compared to the multiple gas phase ones, but there is some concern of maybe we're getting some peptides in here that are not exactly what we think they are because of small differences in sequence. But so far, it's looking really exciting. We're exploring peptide 1, where precursor looks really good. Fragments also look really good. So as we would expect, we're getting a really nice DIA analysis of this peptide. But on the second example here, basically we're missing the precursor, but we're getting the fragment ions. So this is actually an instance where DDA would have fallen down and we would have missed this peptide. But now we're actually detecting it. So it's an instance where DIA is giving us more than we would have gotten compared to DDA. And let's see. And so then we can compare what these look like in vertical profiles going down into the ocean. Now here's some of this DIA data. And these are injection triplicates, giving the error bars here. And they look really great. We're getting some real biological structure with more of these enzymes and proteins in the upper water column, where these microbes are more abundant. And it looks similar to our prior MRM data so far. And here's the DIA versus MRM. And I haven't corrected these for the protein in the water column yet. So this is preliminary here for these comparisons, but it's looking like we're getting real signals of ocean proteins and thousands of them simultaneously with DIA. So just to wrap up, I think maybe I should have renamed the title 80,000 DIA fragment ion intensities for ocean protein sections under the sea instead of the original title. And with that, I can wrap up. I want to thank Matt, Don, in particular, our clinicians who have done the mass spectrometry and the data and the sample extraction, and the Ocean Protein Portal team, and Brian for collaborating with us. And here's the links for the Ocean Protein Portal if you're interested in using it for research or education. And feel free to reach out to us if you're interested in that, and the Metatrip capabilities as well. So thanks very much.
Thank you, Mac. That was a super cool presentation, cool science, and the pictures were awesome.